Thanks everybody for joining me. So I'm just gonna kind of ask some questions for a while. Feel free to jump in if you have a burning answer to any of them, or if it's not relevant to you, you don't have to answer them. So let's just jump right in. Kidneys. So we've been hearing a lot about kidneys this weekend and in cryonics research. Why is kidney research prioritized over direct brain research in cryonics? So it may be obvious to you guys, but to a lot of cryonics members I talk to, they're actually confused why kidneys is the main source of what we do.
Okay, I'll take a stab since I do kidneys. Essentially, I mean, obviously for cryonics, brain preservation is very, very important. But for the overall goal of cryonics to like freeze or vitrify, cryopreserve, and then reanimate an entire organism, there's a lot of steps that need to be done. With organs like kidneys, one of the nice things about them is that they can be transplanted. So you can actually test viability after cryopreservation and thawing. Right now, there is like very little capability of cryopreserving and then transplanting a brain. So, but obviously the research that other people here are doing on the brain is also incredibly important. But like I said, there's a lot of steps to get there. They all need to be done. Some of them sequentially. A lot of them can be done at the same time. And right now our focus is on kidneys, but we're also looking into getting into brain cryopreservation research as well.
And I imagine kidneys are just a lot easier to procure overall.
Way easier.
Okay. I guess for anybody here who's actually doing direct brain research at the moment, if you could speak to why you're doing that over kidneys.
Yeah. So, well, as the name as my lab entails, Advanced Neurobiosciences, we almost do exclusively neurocryobiology, brain cryopreservation. It's only recent when we accepted to do the whole body project that I talked about, that we're sort of venturing out. But yeah, sort of your question is a good question maybe for other research group, but we've mostly doing only brain. Not only doing brain cryopreservation, we also looked at the effects of ischemia on the brain. We also looked at how does the brain respond to ischemic conditions when you do cryoprotection. So yeah, with us, it has been all brain. So, yeah.
Ralph?
So there may be different reasons why you like to work also on the kidneys. And the argument to also work on kidneys is, for example, that society would accept it better. And I mean, it's easier. It's easier to if kidney cryopreservation works, then to say this is a legit area and so we need to also bring hearts back and so on, and then organ banking would be possible, and then a brain would be just another organ. So for society it would be easier to accept. Right. But for the society here, of course, I feel, and that's why I also work on brains, focus on brains. It's most important. So there are arguments for different sides, and whatever you prefer, I think.
So just another perspective, we work on whole organisms. Obviously, when you're conducting research, you need to simplify the problem in some way. One way is to kind of break up the body into the different organs and treat it that way. Our simplification is to work on smaller organisms, but that whole organism. For me, I think a major fear of mine is always that we'll go down a path that will be a dead end and not be able to translate to some later stage that we want to go down. A fear of mine of focusing on individual organs. And take a step back. So my goal would be cryonics of a whole body. So I'd want to preserve the brain and all of the organs together. And a fear of mine would be if you're optimizing one organ, then perhaps that might not translate well to the whole body. So yeah, so we're starting with the whole organism, which will include the brain, does include the brain, but obviously we're needing to simplify, and ours is going small.
And I believe you're starting with zebrafish.
That's right, really small.
And kind of going back to the kidney, would one of the other factors be organ preservation and the fact that there's like a commercial market for kidneys?
Yeah, I think that is a good answer, right? If you look at like, okay, what could this yield in terms of commercial applications? Yeah, brain preservation, not really. I mean, there might be some exotic uses in brain banking and maybe storing tissue for research, but the case for organ preservation has always been a lot more compelling. So that is a reason for it. And maybe that's also one of the reasons why some of the major cryobiology labs have that as a big component, because there's always this expectation like, well, maybe we find something that is commercially viable in organ preservation and that can fund sort of more exotic brain-oriented, more cryonic-centered research, for sure. Yeah.
I think academically, brain preservation research still has great value in like advancing studies in like Alzheimer's and other brain diseases and things. Yeah. So like if the better you can cryopreserve brain tissue, maybe you can kind of sell it as brain tissue is very hard to get. It's very precious. If you can do cryopreservation of brains and then take tissue slices and do research on those for things like Alzheimer's or other brain diseases, that's still pretty important.
So I'm going to keep harping on kidneys for a second. So we particularly rabbit kidneys. So we've been hearing about rabbit kidney experiments for forever. Kind of every time I turn around, I'm seeing pictures of rabbit kidneys cryopreserved. But and I could be wrong, and tell me if I'm wrong, it feels like there's never any breakthroughs. Or at least breakthroughs that really stand out. I guess, why does it take so long to progress in this kind of research?
So this was last week or so, Brian. Last week there was another successful rabbit brain which was vitified and was after rewarming.
Kidney, yes.
Kidney was after rewarming. It was transplanted, and the rabbit was surviving on only that vitified kidney for almost 2 years. So. So it was just recently published. So
okay, and that was different than the previous the previous time, because I don't remember. Yeah, it was,
yes, because it seemed that compared to the first rabbit, this rabbit seemed to be normal, clinically normal, and so yes, it is a big difference here.
And the technology as well, the dielectric, was that also a
yes
progress?
Yeah.
And the time frame on that, what was the kind of time frame between this most recent one and that last one I'm referring to?
The first one was published in 2009.
Okay, yeah.
And this one is So
then I guess I'm curious. That's a huge time span.
Yes, I agree.
And it's just hard for me to wrap my head, because I'm not a researcher, and I'm probably underestimating the difficulty in the situation. And I think for those who aren't researchers, we don't have a good conception of why 15 years would go by and then just a slightly different outcome, better outcome. But yeah, so that's kind of what I'm getting at, trying to understand the complexity here.
Yeah, I'm not part of this program, okay. But I think it just tells me how hard it is to cryopreserve. And when you would do more experiments, it may not help. So you have to do the right changes. So there are so many parameters when you go through the whole cryopreservation process which fail. And so what we want is something which is repeatable.
And would kind of other people agree that perhaps mine and others' perception of this is just not in line with how the actual world of research works in terms of timeframes?
Well, it depends, right? I mean, there are certainly topics that seem to go really fast, and you see a lot of breakthroughs, but there are other topics where no progress has been made at all. One that I'm pretty familiar with is neuroprotectants. If you have an out-of-hospital cardiac arrest or stroke, they do a lot of things. They give you, hypothermia or but there is actually no FDA-approved neuroprotectant. And they've tried thousands of molecules. And some of them we also have used in cryonics because they look promising. So that's sort of not to defend sort of the pacing that you're curious about, but in a lot of these sort of really impactful fields, it's not uncommon, and sometimes there is no progress at all. And I think in this case, it's, again, this is also, I'm not doing kidney cryopreservation, but I have seen sort of the subtle tweaks that were made in cryoprotectant design and loading and unloading protocols, and sort of I get it, but I could see that these kind of changes that they're making are very exotic to the outsider. It's like how to explain that to the general public, like how now we're locking in this slightly lower pressure and we dial down this CPA in the mix by 2%. That's hard to explain.
Yeah, and kind of talking to Nick at Alcor about some of his research, I think wholly underestimating the kind of tediousness and the amount of repetition that has to happen to just move things forward in a very basic way. I guess while we're on the topic of timeframes and kind of things taking a while, I just want to kind of everyone to answer kind of about their own projects and work. What would you say are the biggest bottlenecks for you in whatever it is that you're researching?
I'm not allowed to say funding, yeah.
Yes, and no funding, because that's
everyone's bottleneck.
Everybody's problem. Everybody has that problem. So bottlenecks other than funding.
I'm still learning so much, so it's hard to say exactly what the bottlenecks are, just because I'm trying to get my head wrapped around all of the different research. I still have like 5 books to read from everyone up here. But I think the biggest bottleneck maybe just for the field in general is just we need more cryobiologists and working on the problems. There's not, there's not that many working on it compared to a lot of other things like cancer research, HIV research, all those things. There's just not enough people working on the problem. It takes a lot. Scientific advancement is, based on every time you see a big advancement, it's usually based off the backs of 1,000 other small little steps created by a wide variety of scientists doing work, unseen. And we don't, we could really use a lot more just people looking into the problems and working on it.
I guess any ideas about ways in which we could attract more cryobiologists, or even kind of what do you see as the reason why they wouldn't be attracted to this field? I mean, I guess it's obvious why they might not be attracted to cryonics specifically for the reasons very few people are. But
yeah, and again, it comes back down to, what are the societal benefits of the research, right? And I think we're actually getting more. I think there's more people interested in. If you go to cryobiology conferences, there are a lot more people now than I think there ever were. They're working on tissue and organ cryopreservation strategies and different ones. There's a lot more interest now from pharmaceutical companies working on cryoprotection strategies, because there's a lot more cell therapies that are coming out, like NK cell therapies, CAR T cell therapies for cancer. And, these are biological products that need to be cryopreserved very well. So there's more money, I think, coming into that space. So I think the problem is starting to get better with more people, more money, more research coming in. I think we might be at a crossroads, but again, I'm still pretty new at this, so I'd be interested in seeing what other people think about that.
Unless anybody has something to respond, we can continue with personal bottlenecks in your work.
Yeah, I'd say, and this is sort of the foundation of our company, is that from my perspective, the bottleneck is just the rate of experimentation. So being able to do that as quickly as possible, that's at the heart of why we picked the species zebrafish that we're going to go after first, just being able to experiment very rapidly. Yeah, and so I mean, we're at an early phase, but we are racing, and the immediate bottleneck is shipping of equipment and trying to get to that point where we can be doing experiments rapidly, hundreds, thousands a day. And the hope, our bet, is that that will pay off.
Yeah, I think my answer would be a little bit of an elaboration on the point sort of we addressed earlier about the pace of research. I mean, if you want statistical significance, of course, you need to do a lot of experiments. So there's only so many things you can test. But as I was talking about in my previous presentation, it's not just cryobiology, right? Generally, when you work with whole organs or complex mammalian systems, you need to do cardiopulmonary bypass technologies. I mean, do you ever run into someone who said, "Oh, I have a background in cryobiology and also in small animal CPB"? So bringing all that expertise together, that is non-trivial. And in my lab, we spent an enormous amount of time just trying to figure out how to model something. Sometimes we need to build equipment because there is no mainstream off-the-shelf item that is available for us. And that takes a lot of time. And that's why I'm saying that maybe also sort of answering the question that was asked earlier about the pace. Well, it is because we have to build the physical infrastructure to do it often ourselves. And there are only like maybe really a handful of people in the world who have the appropriate sort of background to do this. When we were brainstorming about the small animal whole body cryopreservation project, we had to see where the experts were currently in the world on small animal recovery cardiopulmonary bypass. And it turned out there were only maybe 2 or 3 groups that were doing that in the world. So one in Japan, maybe one in Germany, if they're still doing it. So yeah, so the things that have to come together for that are clear, but bringing them together and finding these people, it's like really hard. And if you cannot do so, you have to go through a long process of self-education and teaching yourself to do these things, because there's just not that kind of expertise readily available. And I think that's very different, I think, from a lot of areas in aging research where, okay, I know these tools, I know so many of these guys who can do that, right? And I think for our research, especially when you're dealing with whole organisms and organs, that's a lot tougher.
So, what you said before is also true for our limitations in our ultrastructure research, namely the rate of experiments. However, this is not only the only limitation. The one problem is that we have to wait for the imaging results too long. It takes sometimes a month or 2. And during that time, we did a lot of other experiments already. But so it's basically the feedback loop. Ideally, we have a faster feedback, which that then would guide the experiments better. Basically, we would be faster, maybe totally. And there's another area, namely in brain viability testing. There we have a small team only, and the experiments are right now 11 hours around. We would need to work maybe 15, 16 hours, and that's a simple reason, because the brains sometimes need to be perfused at least for 3 hours, at least in the simplest case, to get EEG signals back indicating viability. And we did one test, a very simple test, where we perfused for 5 hours, and the EEG signals were very well visible, indicating viability. So, but we have to extend the experiments in terms of time, and this, it's just a lot of burden on a small group, and we have to. I think we need more people to be able to do shift work, for example.
So I'm interested in structural brain preservation using fixatives for the reason that, notwithstanding Ralph's new results, it's the method that's been published that best shows retention in a good way of like ultrastructure of the brain and connectivity with a way in which it can be traced out. Now, that's good if you're concerned with like holding on to synapses and other things that we think retain memories. But the difficulty with doing preservation through these only structural fixative methods is you can't just warm it back up again and then test whether it's viable or has electrophysiological properties that you expect, or even do some sort of memory test on the animal. So you need some sort of proxy measure for like, is this going to work with the mind of like maybe one day being able to use that to do emulation or uploading or use nanobots to revive it. And it's no good to just like hope it will work at some point in the future based on theoretical arguments that we won't be able to test for years and years and years. What would be good is to have, milestones that can give you some sense of like, well, we can't do the revival yet, but we get increasingly confident that that might be possible. Or on the other side, to show you that like, actually, no, your method isn't working. And the things that are demonstrations of that are either doing things like taking a preserved brain and being able to decode particular memories out of that structure, or even to be able to do like full emulations of not necessarily human brains, but smaller animal brains. So zebrafinch or C. elegans or flies or any sort of like smaller animal. Luckily, there is actually progress being made in this sort of field. So for example, in October of last year, there was the first publication of the full connectome of a fly brain. And there was also, I wouldn't call it an emulation because it wasn't a perfect causal replication of the fly brain, but a very, very good simulation that starts to approximate an emulation, which showed some of the same behaviors of the original fly. So for example, they like took the digital neurons, stimulated the sugar sensing equivalent neurons, and saw activation of the motor neurons in the digital model corresponding to the fly trying to like stick out its proboscis and tongue. So basically what the bottlenecks are just like research groups working on these projects, like trying to do the full connectome of a mouse, for example, is being worked on at the moment, and trying to do better emulations of mouse brains, or alternatively trying to do memory decoding from a preserved animal brain. So for example, there's a prize out at the moment for any research group that manages to show the decoding of a non-trivial memory from a preserved brain. That might be something like, you have a mouse, you train it on a particular maze navigation, and then later on you can take its brain, image it, and show that like, oh, I know this mouse took this particular route, or something to that effect. That's what we want to see more work being done on. Basically, these proxy measures to give you a sense of whether the fixation style preservation is actually holding on to the things we care about.
Okay, thank you. So I'm going to shift gears to rewarming. So I guess someone who feels confident in explanation, first let's just start with why is rewarming harder or riskier than cooling? So what makes the physics and the biology of warming more challenging?
Well, I mentioned this in my presentation already. This was published and mentioned in the journals of cryobiology. Peter Maser had basically a paper about why the warming rate is more important than the cooling rate. And the main reason is during cooling, we pass first through the let's say we have high concentrations of cryoprotective agents during we want to vitrify something, a sample. Then we pass the temperature region of crystal growth, and if there are no nuclei, small ice crystals in the sample, then nothing can grow. But this is for M22, it's around minus 60 degrees Celsius. But when we cool further down, then we're reaching a region around the class transition temperature. For M22 is 1 123 degrees Celsius, where the nucleation rate is maximum. But everything is very viscous, so it is not very likely that nuclei can occur. And so this means practically during cooling, ice formation does not occur during vitrification. But then, if there are small nuclei, a few nuclei in the sample, then during rewarming, they can grow into ice crystals. And like I mentioned in the presentation, we have to outrun this ice crystal growth. So, but this is not only true for vitrification, but also for freezing. So.
I think usually the critical warming rate for any CPA is something like 3 or 4 times greater than the critical cooling rate. So you got to warm a lot faster than you cool to prevent the ice crystal formation.
So,
I'll just restate, because I think that was a fantastic detail, but for the high level, I was just trying to explain it to my wife last night too, that that when ice is formed, it needs to nucleate and then it needs to grow. And these are sort of stochastic events. The region in which the ice grows is, usually where there's low viscosity, so that new water molecules can join on really easily, and that's kind of higher. And then when you're at a lower temperature, that's when the nucleation is more likely to happen. So when you're cooling, it's not that big of a deal because you're going through this phase where ice could grow, but there's not very many nucleation events. And then you get into the nucleation events, but they can't really grow because it's high viscosity. When you're warming, you're facing a much harder problem. You're going through a region where nucleation events can happen, and now you're going to a less viscous region where now those nucleation sites can now grow really rapidly. So the warming is a harder problem.
Yeah, well, actually, my answer to this can be pretty succinct because the vitrification solution we're using in our lab most of the time, VM1, which is also used by Tomorrow Bio and Cryonics Institute, in the concentrations we use it's basically sort of unfreezable. It's actually you have to do a lot of stuff to it for it to freeze. That probably will come at a toxicity cost. Like, I'm not aware of VM1 ever being used for, mainstream organ preservation like the kidneys or whatever. So that's a reason there's a less of a problem for us. The other reason is just our research model so far has mostly been hippocampal slices, and you don't have that scale, that size issue. So we can get away with very sort of straightforward sort of warming methods. But if we're going on this path of whole body cryopreservation, it's going to be very important for us as well. And these are serious challenges for sure.
Now, in terms of rewarming methods, so I've heard a bunch of them. I'm just kind of trying to wrap my head around all the options, but you have like nanoparticles, ultrasound. I guess I'm curious from all of you, which methods do you think are the most promising, and the ones that you, I guess, intend to work on, and which do you think, if any, are might be dead ends? And anybody can start.
Well, my answer is going to be very succinct. I'm going to probably just defer to my esteemed colleague Brian Woke. Whatever he thinks is the best warming method, I will take it advice and just roll with it instead of agonizing myself over it.
Okay. Can we get a mic to Brian Woke?
Yes, I agree. Brian is the expert on this. Brian Woke.
When all you got is a hammer, everything looks like a nail. So I built a dielectric warming system.
Yeah, so I know nano warming and dielectric warming kind of competing when we want to rewarm organs like kidneys, and there's research in both areas continuing. Both are legit, and I think dielectric warming has great potentials. I think for larger organs like like human kidneys even.
Yeah, I think it's still pretty early days for both methods. So I think there's it's one of those things. They both look promising. Who knows? I think there's a lot more work that needs to be done to figure out. There may be some cases where one is more advantageous than the other, but we don't know.
And does anybody have opinions on the ultrasound warming? I don't know much about it. What are those like nods of disapproval or nods of?
No idea.
I don't know.
Yeah.
Okay.
No idea.
So I just want to step back, say something about the dielectric warming. The advantage is in general that the wavelength is relatively large, and it's about, we want to homogeneously rewarm the organ, and this works for dielectric warming. For ultrasound, I'm not sure, to be honest. That's
So I guess we'll go on to a topic that I feel like there's constant debate about. So hopefully we can have a little debate about it right now. But cryoprotective agents.
Oh, no.
So actually, before we get into the rough and tumble of it, what, if any, progress is being made in developments to new cryoprotective agents? So. Is there anything on the horizon that's a paradigm shift, or is it just very slow iterative processes?
Yeah, I can take this question. So, like I mentioned also in the presentation, M22 was designed over 20 years ago, and in 2004, in one of Greg's papers, he was reviewing why, or basically the history of why we came, our company came to this high standard. I think there are different. Are there 4 generations of cryoprotective agents? 6 generations. So. So I know there were a lot of attempts to top this, and it turned out that M22 is just was the best. So that's one answer, but we don't stand still. I can tell you this. So I cannot give you the details, but we don't stand still.
Okay. Well, then I guess if you have a thought.
Yeah, I can. Yeah. One thing I've learned is to really come to appreciate sort of how good the existing sort of agents are and how hard it is. Like even smaller tweaks often fall flat. And both VM1, the agent that sort of I work with a lot, and M22, it's like there's not a whole lot of change, but it's very sort of hard to do. There was a brief moment, and I think I mentioned that earlier, where I thought, oh, there are these CPAs that can cross the blood-brain barrier, so you don't have to screw around with all these blood-brain barrier openers. And I was very excited, and I think we researched that for at least more than a year. But then we started increasingly realizing that these CPAs that could cross the blood-brain barrier so well were a lot more toxic. So that was sort of the end of that road. Right now, I cannot sort of talk in details. We have seen for the brain specifically that we can tweak with the carrier solution of the cryoprotectant in ways that give you better results. But these are tweaks. They're not true shifts in paradigm. I think that might be possible. A lot of people talk about using computational technologies, for example, to identify new cryoprotectants. That's, I think, not really a capability I have, but I'm in contact with people who are interested, and I'm more than happy to run sort of these cryoprotectants through our models if people say like, okay, this is really great.
I think you're sitting right next to someone who's probably working on this right now.
Yeah, so 2 things. Speaking with Greg Fahey recently, he indicated that he does have something new coming out and that he's excited about. So we'll all be hopeful for that. And then in speaking with the cryobiology community, so the people working on the organ transplants and cells and whatnot, I've asked this question sort of explicitly, like, is it that we've looked and there's nothing better out there, or that or something else? And their general conclusion has been that it's just that we are a very small community and we haven't looked, but the hope is that there are better things out there. I mean, I don't know how to reconcile that with the idea that we've been trying to improve M22, but I mean, maybe the connections, like the interactions of these CPA components and the idea that two toxic components can interact and become less toxic than either one of them combined or either one of them alone, and I think vice versa. So there may just be interactions that we're not smart enough for, and the hope is, at least with my company, that AI might be able to help us stupid humans out with this.
So regarding M22, I think the use on brains, at least I can tell, there are many more experiments we need to perform to characterize under which conditions there are good outputs. It's not, toxicity is one of the main issues we have to overcome, but it's not only it. The cryopreservation process is very complex, and there are a lot of an enormous amount of parameters involved, and when we have not optimized one parameter, we run into issues such as I showed with the white spaces, and so which is maybe independent of M22 or what vitrification solution we use. So we have to do a lot of more experiments to optimize.
I guess for now, M22 seems like the head honcho, but on a practical level, how different is M22 to its closest competitor? So Alcor uses M22, and they insist that it's worth the additional expense. But some of the other orgs, CI and Tomorrow, don't think the trade-off is worth it. So I guess what would you guys say that the trade-off is between that in your experience?
Yeah, I think I can answer that one sort of pretty well, at least in the sense that I've worked a lot with VM1, which is sort of the other agent. I've worked with M22 as well. M22, like much more wilder things have been done with M22, right? So as I was saying earlier, it has been used for organ cryopreservation and then transplants, and then sort of, you have long-term viability. This has never been even sort of tried with VM1. What we do know is when you look at the hippocampal slice viability, Europachugan did that. There was only a very small difference between M22 and VM1, and Ryan or Ralph can correct me. I recall it's not even the case that M22 is necessarily the best 21st century medicine agent when it comes to central nervous system viability testing. But I think when you talk about the cryonics context, are these differences meaningful? I don't know. If a patient, if the brain is very ischemic and it has basically no viability anymore, does it make a big difference that one agent has a slightly higher viability in controlled lab situations, and some people might say that's not so important. If we're still really shrinking the brain, and some of these agents that are so good about M22 cannot cross the blood-brain barrier, is there an advantage to using them? I think these are sort of all reasonable questions. I think right now, one thing I think that has become more of an interesting talking point, because the field is moving towards field cryoprotection increasingly, and like with Tomorrow Bio, whole body field cryoprotection is their universal sort of procedure. It's like, what agent is best suited for that? And then you want an agent that is sort of almost unfreezable, whatever you do to it, because you might have a patient that is in dry ice without any further cooling down for maybe several days. So that may change sort of the perspective, what agent sort of is ideal. But I think sort of they're both good agents. It's I think one benefit of M22 is you can point to this literature and say, oh, look, a kidney has been reversibly cryopreserved, which you cannot do for VM1. I'm not saying it's not possible, but that sort of hasn't been tried yet. I'm working on a paper, actually, we're pretty close, showing viability for hippocampal slices, and even after loading and unloading of the brain. And I think VM1 looks sort of pretty decent. So that's sort of my nuanced take on it.
I hope you'll forgive me, but I want to ask them a question as well, which is, do we understand in principle both the physics of CPAs preventing ice and the physiological mechanisms of toxicity, such that in principle we could do rational sort of CPA design? Or are we stuck with, we don't really understand it, we just got to try different methods, whether it's like AI and experimental high throughput? Like how well do we understand, how much can we narrow the space down just based on theory?
So I think we understand a lot. The different components of the vitrification solution have different tasks, like lowering the freezing point, like they need to be stable and prevent nucleation, of course. Then there are some polymers in M22 in this case, which adhere to the surface of ice crystals and prevent them from water molecules to attach. And I think we know quite a lot about how they act, the different components. And there was a recent paper coming out which showed from Adam Higgins showing different combinations between, it was basically a high throughput study. And they basically found out that some combinations of cryoprotective agent components work very well. So I think, yes, there can be theoretical studies. I think high throughput studies would be also interesting. If they are then finally better than M22, that is not clear. And maybe I'd like to, regarding the comparison between M22 and VM1, first I have to say I'm possibly biased, but because our company is selling M22. However, first I'm a researcher and not a salesman. Second, I can say the company is not dependent on the revenue of M22. But Ashwin is right when he says that most of the publications, successful vitrifications of brain tissues and organs have been done with M22. So M22 is in the literature, and this, I think, is important that we can have a basis, a good foundation, a scientific foundation for the cryonics community to be legit. I think that's important.
Yeah. And Mark, I know from the podcast you're doing kind of all these different variables and for what you're looking for cryoprotectants to do are kind of very important for your model. Could you expand a little bit upon the types of variables you're looking for and how different CPAs kind of act?
Yeah, I'm not sure that I have much to add there. I'm fairly inexperienced with it, but yeah, it's just we will be optimizing anything that we can there. So cooling rates, if there's any annealing, if we want to do, try to elicit some heat shock protein generation, so we can play with the temperature profile, and then we'll also be optimizing the compositions of the chemicals. I don't think that I have more to say than that. I mean, I would love, this is sort of a call out. We don't have the funds to be doing this, but just being able to have a large data set of physical properties and cryoprotectant components put together. So, viscosity at different temperatures. I supposedly, and this is to answer your question, Ariel, too, I guess glass transition is something that you're able to predict well from the glass transitions of each of the components, or something they compose in an easy formula. But yeah, so I don't have much to answer other than we're going to try to optimize everything.
Moving on to, I guess, perfusion quality. I guess what are the best ways? I mean, we know CAT scans, but are there any other additional ways to evaluate perfusion quality? And kind of a similar question to before, are there any paradigm shifting improvements on being able to measure that? Is it even needed, or do we have good understanding with the current technologies of how to how to address the quality of perfusion?
Oh, okay. I mean, obviously we started with doing CT scans, and we're trying to see if we can look at, how good vitrification is if we do pre and post cryopreservation CT scans. There's work to be done that I would like to see if we can use that method to better quantify, after doing some like scanning calorimetry and those things that guys at 21CM do, and compare that to like density changes from the CT scans, and maybe we can get a little bit easier and better way of looking at cryopreservation quality that way. But it's obviously we're still just in the infancy on seeing if that's even a viable thing to do. Other than that, I don't have any. I would love to hear some good or better ideas. Like, I'm sure Ralph knows a lot more than I do about this.
Yeah, I can say something to that. I mean, in our case, we try to work under ideal conditions, meaning we don't have issues with warm or cold ischemic injuries. So this means that our perfusability is very good, and this is an ideal case, which is for a chronics case not usually.
So, aside from CT scans, there are always very simple indicators whether you're perfusing well. It's like getting a good flow rate for a given pressure. If there's a lot of ischemia, it just really slows down. Weight gain for whole body cryopreservation. If there's a lot of weight gain, like way above and beyond what you would typically see, that's a good indicator that there's a lot of ischemia. And that, I think, this is something we haven't really sort of talked about, but to me, I think it's really important. And when I started my lab, I'm like, how can I possibly do anything meaningful or compete with these guys from 21st century medicine? I'm like, okay, let's research how to do cryopreservation, cryoprotection of the ischemic brain. And if you would ask me, so what is sort of the toughest problem in practical cryonics research, that would be the one. I've thrown so many different approaches at it, like, higher pressures, adding thrombolytics to the cryoprotectant, higher viscosity protectants. What we have even tried to, decompressive craniotomy to allow the brain to swell. And we have never been able to crack the problem of getting really good global perfusion in a ischemically compromised brain. And a lot of cryonics patients have ischemically compromised brains. So that is a very, I think, unsexy topic, even among sort of cryonics researchers, but it is of extreme importance. But I haven't seen much that shows a whole lot of promise. But, our lab getting the micro CT scanner, maybe there are new ways to look at it. But when we have just a control brain, no ice formation, everything is good. That's pretty easy. The brain is very well vascularized. No ice formation. It's really with the real world application, the ischemic brains, where things go really awry.
We've heard quite a few times about static CT images being taken post preservation procedure from Tomorrow and from Alcor. One thing I would point out is that, for example, if you took a patient who had a stroke and you wanted to do CT imaging, what's also done is something called dynamic CT, where you can measure like the ongoing level of like blood flow through a brain region. And you could do the same dynamic sort of CT imaging in theory during the like perfusion steps. And there's at least, I know, some interest in trying to do this dynamic perfusion imaging during like actual preservation steps to see if you're getting good flow before you just look afterwards and you see whether there was ice formation or not. And potentially that could even be used to indicate that an intervention might be necessary if some area isn't being perfused well during the preservation procedure. Then the other thing, obviously, is you can always do biopsies afterwards as well. You take a sample out, you look at it, you see whether it's been preserved well or not.
Does that dynamic route require specialized equipment other than the current CT scans, or is it more of a technique?
I don't know which exact CT scanners can do dynamic, but it's definitely a thing that many CTs can. Yeah.
As regular CT machine can do it, just the modality you need a contrast.
Okay.
I think, do you always need a contrast? I don't think you necessarily. Okay, all right. I defer to Boris.
We've done dynamic CT scanning during multi-hour environmental conditions. It's quite sitting out. We do not use contrast because if you add contrast, then you lose the ability of CT to quantify its CPA. You basically contaminate it. Regarding the biopsy issue, if you take samples region by region in an organ or in the whole body, then the gold standard quantification of quality is differential scanning tolerance. Where you measure the melting point of each solution sample. If we need to go one step beyond that, I define something called photography FLC. With that, you can actually measure the concentration of each individual component of a multi-component solution like FLC2. And since I'm up here talking anyway, I'll just mention as a correction a statement that some CPAs don't cross the blood-brain barrier. By definition, all penetrating CPAs cross the blood-brain barrier because if they're penetrating means that the cell is penetrating the cell's line of blood vessels will go in just slower than other organs that have gaps between these cells. And we've actually measured that in FLC. You can see the individual ingredients of FLC2 on the other side of the blood-brain barrier, just there at a lower concentration, like in their
We need to get another chair for Brian.
Yeah. Any other thoughts before we move on?
One thing I wanted to mention, maybe you're going to ask a question about it, like sort of traditional cryo versus sort of chemical fixation, but sort of these chemical fixation guys, based on sort of my conversation with them, running in the same issue of perfusion of sort of the ischemic brain, right? Because the brain is very ischemic. You also have poor or incomplete uptake sort of the fixatives. So in that sense, we're sort of in the same boat, sort of the traditional cryo guys and the chemical fixation. There might actually be some arguments why people who use CPAs are in a slightly better spot, but it's maybe sort of maybe an exotic argument. But I just wanted to mention that when it comes to struggling with perfusion of ischemic brains, that applies pretty much the same sort of to the, the chemical fixation approach, at least when you use perfusion,
So you mentioned the ischemic brains. What's the damage that's happening? Is it at the membrane level, like they're closing off and they're not able to come in, or is it blockages or something?
I think it's multifactorial. Often red blood cells basically sink to the bottom of the vessels and it becomes hyperviscous, and then you start perfusing. So the perfuse takes sort of the path of least resistance. So that's one. Then you have energy depletion in the cell, which causes swelling of cells and also of endothelial cells. Then the vessels themselves become really leaky. So you're just perfusing, and it goes everywhere, but not in a good way. So there's a variety of things, but it's not only blood. Sometimes people think if you just give heparin, you don't have that issue, and certainly it's greatly reduced. But then you still have the energy depletion and the swelling bit and the leaky vessels. Like for example, if like classic, or maybe I should say now cryonics protocol was to do a blood washout and then ship on water out to the cryonic facility. But if that whole procedure takes 24 hours, you can have reasonably good perfusability, but you get a lot of edema because the endothelial cells have become very leaky. So these are some of the things that go into that mix. Yeah.
How are we doing on time? Maybe 5 minutes? Okay, so I'll ask a couple more questions and then we'll open it up to the audience. So research priorities, that everybody can answer this. If you could direct significant new funding, no strings attached, to one priority within research, something other than what you're currently doing, what would it be? You need to take a second to think about it. That's fine.
I don't know. It's the other than you're currently doing throws me
Yeah.
off because
Yeah.
I would just want more money.
I know, right?
Like for example,
Organs.
like to me, being able to perfuse most cryonics patients really well, even if they have significant ischemic compromise. To me, that is kind of the holy grail of practical cryonics, and just sort of the more sort of funds and resources would be invested in that, the better, especially because it's such a sort of unsexy topic, sort of even within our field. So that would really be, my number one thing that I think needs to be much further explored and funded.
Characterizing neuronal traceability in ideal and non-ideal cases, just to try and get a good sense of what is actually going on ultrastructurally in various ischemia conditions, various fixation conditions, various just cryoprotected conditions, so that you had really good metrics to compare just how bad or how good things were under all these different circumstances.
Probably, I know we're doing pig kidneys and whatnot, but I'm still, I really want to see more research done on, the issue of whether or not the shrinkage you see is actually, when you cryopreserve a brain, is that important or not? Those kinds of studies, looking at, how does that affect viability? Doing cryopreservation, see the shrinkage, take a, sample, see if it's, still functional, or, doing that, unloading it, see if the, brain swells back up and see if it's functional. Those, the step-by-step studies needed to show how much of a problem is that, or is it a problem, or what can we do to mitigate that? I know there's some work being done on that, but I'd like to see a lot more on it.
Yeah, I guess I'd say inexpensive, rapid measurement. So something where you don't have to wait, days or whatnot for the imagery to come back, but different measurements. And then this would help science in general, I think, but making it more accessible to the individual scientists to get results quickly of their experiments.
Yes, I can fully agree what you said. It's true for us as well. As I mentioned before, the feedback loop should be, time was shorter. So, but besides that, since we have a small group and 3 different projects right now in my team, we are working in parallel because we like to use synergy effects, so we can learn from the different projects. And what we are not focusing too much in, blood-brain barrier investigation, we apply. SDS, which was mentioned before, but we could open another path for studying, which then would expand the scope, basically.
Thank you, guys. Okay, let's open it up to the audience.
Yes, thank you. This question is for Ralph in regard to the time frame for getting imaging results. I'm wondering why it takes so long, especially since if I get an MRI, for instance, even as an ordinary patient, I could get that result in less than 24 hours. So what are the sources of the delay?
The sources of delay is, one reason is that the person, we have a department, especially for imaging, it's one person, and this is a shared resource, basically. So it's not only data from or brains from our group, but also organs from other departments. That's one reason. But in principle, the processing takes time. It's not so that this person is working too slowly. So no, but there are but there are different reasons why a delay can occur. That something is failing, and then we cannot get results for a month or many months. It happened before, and so. Yeah, it's a combination between maybe it's funding, because maybe we should, if we have funding for outsourcing it, that would possibly enhance it.
Thank you guys, appreciate it.
Thanks, Daniel.